Hi, I am Aniruddha Panda. So today I'll share a story about protein-lipid interactions, particularly spatial interactions in the membrane and how it is important in the neuronal communication. So in the neuronal communication, so basically you have presynaptic membrane filled with neurotransmitter filled synaptic vesicle, tiny vesicles, and then that fuses with the plasma membrane in some millisecond time scale, and they release the neurotransmitter which then uptaken by the next receptor in the postsynaptic neuron. And this is a most fundamental aspect of any kind of neuronal signaling or chemical synapse. So now if you look into the protein machinery which drives this fusion, you will realize that there are a set of snare proteins. For example, in the vesicle side, you have vesicle associated membrane protein. And in the plasma membrane side, you have a heterodimer of SNAP25 and syntaxin. And these VAM2 and T snare then fuse together to form a 4-helix bundle. And when it's zippering from here to this end, what it does, it's bring the vesicle close to the plasma membrane. And then ultimately that zippering force actually overcome the fusion barrier. Now, the important question in the field is that if you purify these proteins and set up a fusion setup in a test tube in a laboratory timescale, they actually take more than seconds. But in the physiology, the fusion happens within some millisecond timescale. So there are possible hypotheses around hierarchical organization of proteins which are involved in this ultra-fast fusion. And these hypotheses are mostly driven by different set of proteins which are present in both in the plasma membrane side and in the vesicle side. But for the interest of today's talk, I will mainly focus on the protein-lipid interaction because it has also been hypothesized that this protein-lipid interaction also play a crucial role in organizing this snare machinery. So as you now understood that everything is happening in the membrane, so study these protein-lipid interactions, you need a tool set which can actually have a dynamic molecular detection range. So which can have the capability of detecting megadalton protein complexes to all the way few hundred Dalton lipid molecules. And then once you detect the complex, you have to have an ability to deconstruct them into its molecular entity for unambiguous identification. And most importantly, you have to have the capability of detecting these complexes directly from the bilayer. So I have taken mass spectrometry and particularly native mass spectrometry approach to achieve all these goals. And I have developed a platform using liposome where I can reconstitute the protein of interest in a liposome. And in the liposome, you can fine tune the lipid composition, membrane fluidity, membrane curvature, membrane tension, and the protein copy number to mimic any target physiological membrane. And once you prepare this proteoliposome, then you can infuse it in the mass spec. And with the help of supercharging agent, you can actually ablate the protein bilayer in the front end. And as you are actually detecting the protein by ablating it in the front end, your quadrupole is free and you can isolate any protein lipid complex in the quadrupole and then bring it to the collision cell where you can do MSMS experiment, top-down experiment to unambiguously determine the lipid ID. So in a nutshell, this method actually helps you to determine oligomeric assembly of protein directly from membrane. And it has the capability of ID the bound ligands or lipids. And once you ID the ligands or lipids, you can then go back to the tunability of the platform and modulate that particular lipid, and you can see what effects it has in the protein lipid organization. So I applied this in the neuronal synapse, and what I did is that first I reconstitute the vesicle associated membrane protein in synaptic vesicle mimicking liposome by exactly mimicking all the parameters of synaptic vesicle. And then when we applied this method, we saw that there are some satellite peaks associated with the proteins. Now, as I told you earlier that you can isolate one by one, and then you can dissociate them in the quadrupole, and using high resolution mass spectrometry, you can ID them. When we did this, after all the series of peaks, satellite peaks, we realized that they are actually phosphatidylcholine and cholesterol. So in the bilayer of synaptic vesicle, VAM2 specifically binding PC and cholesterol out of all these different lipids present there. So interestingly, if you mix this set of lipids and proteins in detergent and you apply the same method, the specificity is gone. Now the VAM2 is binding all the lipids present in the detergent solution. So that tells us that the specificity of PC cholesterol binding is coming from membrane. So at this stage, we have a fuzzy picture that it's not a individual bimolecular interaction, but it has to do something with the membrane. Now, if that is the case, then if we replace any of the component in the membrane, then that should abrogate the binding. So what we did is we replace all the PC with a ethyl PC, where the phosphate backbone is linked with a ethyl group. And when we did the same experiment, we don't see any PC binding. And most importantly, in this experiment, we don't see any cholesterol binding, although the same percentage of cholesterol is present there. So that actually tells us that there is a link between PC cholesterol and the protein, and there's a tripartite interaction. Now, if that is the case, then that should be also true when we modulate the cholesterol amount. And when we did that experiment, as you can see, from 0 mole% cholesterol to 44 mole% cholesterol, the PC binding increasing linearly. So there is a correlation between PC cholesterol and VAM2 binding in the membrane. Now, in biology, whenever there is a specificity, there has to be some functional trait associated with, because in that specificity actually arises from the evolutionary pressure. So to study that functional trait, we did a simple bulk fusion assay where we put the VAMP in the synaptic vesicle and we put two lipid dye, one is NBDP and one is rhodamine P. Now when they are in the close proximity in the same vesicle, they quench each other because of the overlap in the emission spectra. But if you mix this vesicle with the T-snare vesicle, then they will fuse, and because of fusion, they will be distant apart and you will start getting fluorescent signal. From this fluorescent signal with time, you can quantify how much fusion is happening. So when we applied this in the 100% PC, like if you take the PC as the 100%, you see that in the EPC, where the VAM2 doesn't bind the EPC, you see that 60% abrogation of fusion rate. So what happens to the cholesterol? In the cholesterol also, you can see from 0 to 44 mole% cholesterol, the fusion is linearly increasing. So this is actually then tells us that the PC and cholesterol binding to the protein is important for the fusion. Now, what happens to the thermodynamic stability of the protein in the membrane because of this lipid association? So to study that, we then collaborated with Brandon Rotolo in University of Michigan, and we applied ion mobility mass spectrometry. And it's a very simple assay where you can actually apply energy to unfold the protein, and you can then measure the time it takes to reach the detector, which is the drip time. And then you can do the same experiment with the lipid bound protein, and then the difference between these two actually will tell you the thermodynamic stability of the protein. So Dr. Ileana Lewis, he was a grad student. He did this experiment in Brandon's lab. And as you can see that indeed the VAM2 PC bound, the PC bound VAM2's thermodynamic stability is indeed dependent on the 44% cholesterol. In fact, that is the highest stability. And if you look into the composition of lipid in the synaptic vesicle, 44 mole% is the native synaptic vesicle cholesterol amount. So now it actually tells us that when the PC bound VAM2 is present in the high amount of cholesterol, it actually imparts thermodynamic stability. So now these are all fine. These are actually still now actually we have an idea that there is a tripartite complex formation going on. But if that is the case, then we should visualize it. So then we collaborated with Frédéric Poncet in University of Paris, and we flattened that proteome vesicle into a suspended lipid bilayer. And then in this case, we actually added a little bit of rhodamine PC in, which is the red channel, and the VAM2 is in the green channel with the GFP fluorescence. And as you can see, if you don't have any cholesterol, there is a homogeneous membrane. But the moment you add 30% cholesterol, you can see this puncta formation happening. And if you increase the cholesterol, what is happening is the number of the puncta is remaining same, but the size is increasing. And that we can quantify how much pixel each puncta contains. So in 44 mole percent cholesterol, you have the highest size puncta formation happening. Now, if this is true, then as I told you that at this point we have a proof that there is a VAM cholesterol PC tripartite condensate is going on in the membrane. Now, if that is true, then if you remove any component, then that should destabilize this tripartite complex. So that is the set of experiment we did next. This is the native 44% cholesterol, and where if same experiment when we did without the protein, you don't see any of this puncta formation. And then in the next experiment, this 44% cholesterol bilayer, we added cyclodextrin. What cyclodextrin does is scavenge out the cholesterol. And when you are scavenging out the cholesterol, almost like a movie, you can see that those puncta are now disappearing from the, anyway, so believe me that these punctas are disappearing from the membrane upon. So that tells us that so this is indeed the three component has to come together in the membrane to form this organization. Now, as I told you that, so it started with the native mass spectrometry by just simply observing that the protein is specifically binding to these lipids. And then slowly when we added component experiment, this picture came in that they are indeed in the membrane, they are forming this spatial clustering. Now, as I told you that in the native synaptic vesicle, the 44% is the highest amount of cholesterol. In fact, possibly you cannot create any membrane more than 44 mole percent cholesterol. That is the highest possible level. So what I did, I was wondering, like, how can we manipulate the amount of each of these component? And then what I did next is that I increased the protein copy number. And so when I modulated the protein copy number, so the data is not coming because of the problem here. But what I observed is that if you create lipid to protein ratio of the protein proteoliposome in 400 and the vesicle size is 40 nanometer, that actually gives the optimal fusion. And if you calculate how much protein will be there, that is 77. And there is exactly 70 copy of VAM2 present in synaptic vesicle. So that kind of matches the native synaptic vesicle. And with all this, what we came up with, again, I cannot show you the entire model, but the idea is that when you have high copy number of protein in the synaptic vesicle, it doesn't matter whether you have cholesterol or not. It will fuse faster in the anyway. But when you have 70 copy per vesicle, that is a low copy number. So then you need the cholesterol in the membrane to organize the VAMP in a particular direction, which is orient the VAMP in the T-snare, and that kind of bringing lot of VAMP and T-snare in a spatially cluster organization. And it's kind of, you can take an analogy of breaking a piñata, where 5 people are trying to break the piñata at 5 different places versus 5 people are hitting at the same spot at the same time. So that kind of gives the force of this ultra fast fusion. Of course, there are other proteins involved in it, but this is what the VAM cholesterol does in the membrane. So with this, I would like to take questions, and I'm sorry for that.
Okay, let's take questions.
So you're able to do these reconstituted fusion assays with the liposomes?
Yes.
And when you do that, when you have these snare-mediated fusion, do you distinguish between like what people call as hemi-fusion, kiss and run, and full fusion events? Can you?
No, it's not possible in this setup, but we have a TARP microscopy-based setup where you can monitor individual vesicle. There we can actually track which one is going for kiss and run, which one is going for normal fusion.
people do see these different.
Yes, they are in that setup.
Does it give you the 1,000 times?
Yes, it's definitely 1,000 times faster
Really?
than you.
But when I thought we heard last week, we heard
Yeah.
your
but-
colleague
yeah,
from Jim Rothman's group,
Yes,
and he talked about a six-fold
Symmetry,
six Munk, no, Munk proteins.
Monk 13,
Monk
yes.
13, yeah.
Yeah.
And
So
that-
basically what actually, I just showed about only the VAM2, which is in the vesicle, and I talked about, but there is synaptophysin from the vesicle side, which actually has six-fold symmetry, and one synaptophysin bind to two VAM2. So that actually 12 VAM facing the plasma membrane side. From the plasma membrane side, you have Monk 13, which again forms a six-fold symmetry and orient the T-snare in such a way that the VAM from the vesicle and the T-snare from the plasma membrane, they are exactly in close proximity. But what my data is suggesting here today is that the cholesterol is already, before synaptophysin comes in picture, cholesterol is already bringing all the VAM together in a spatially clustered manner, and it's now ready for the synaptophysin to, organize into six-fold symmetry. And most importantly, these accessory proteins, synaptophysin, Monk13, these are post-trafficking protein inserted in the synaptic vesicle. So you need some kind of arrangement before they come into play.
Okay, let's thank Aniruddha again.